Did you know there's a robot army that forces cells into single file and blasts them with lasers, splattering the results onto a graph for doctors to read? It's called flow cytometry. Here's the wild story of how we got here. Quick key before we hit the gas. Picture every cell as a car, and the whole flow cytometer as a high-tech superhighway built to inspect them. Every breakthrough ahead is just a smarter way to handle that traffic. It starts on a microscope slide. For centuries, we could only study cells one at a time, like trying to identify every car on a superhighway while parked. Then, in 1742, Lomonosov saw that particles scatter light, the seed of every modern detector, 1953. What if you made each cell drive over a tiny scale? In his Chicago basement, Wallace Coulter did exactly that, using a dip in electrical resistance to weigh cells by volume. It gave us the automated blood count, and hematology was never the same, but a highway is chaos. The fix? Construction cones. Crosland-Taylor wrapped the sample in a faster sheath fluid, pinching every cell into a perfect, single-file, 10-micron line. Suddenly the clogging was gone, and true flow was born, 1965. Now imagine teleporting only the red sports cars off the highway. At Los Alamos, Mac Fulwyler borrowed inkjet printer tech, slapping a charge on droplets and steering them with 12,000-volt plates. The first cell sorter. Then we painted the cars in glowing neon. Gourde built the first fluorescence cytometer, and Herzenberg added lasers and coined the fax CS. By 1974, it was commercial, and you could finally see exactly who was driving. 10 colors of neon paint is dazzling until the colors bleed. Rotor's tandem dies push the parameters higher, and Bagwell's compensation math used linear algebra to unmix the spillover. Flow became as much software as hardware. This is where it saves lives. Doctors use flow to find a single leukemia cell hiding in a city of normal traffic. The ISHAGE protocols gating standardized stem cell counting, making transplants dramatically safer. Why squint through colored filters when you can photograph the whole rainbow? Spectral flow, theorized by Robinson and perfected by Cytech, captures each cell's full spectral fingerprint, then unmixes it with math. 40 plus parameters, no problem. Then we ditch paint entirely. Mass cytometry tags cells with heavy metals, vaporizes them in a 5500-degree plasma torch, and counts the atoms by mass. 50 plus parameters, with essentially zero background. Beautiful, and a little terrifying. And today, the highway runs itself. Lab-on-a-chip hardware meets artificial intelligence, with algorithms that find patterns across 50 dimensions no human could ever see. From one scattered photon to a 50-parameter data stream. That's the history of flow. Whether you're a lab leveling up your training, a trainee prepping for the boards, a future partner with big ideas, or simply curious about laboratory science, CruxSci was built for you. The timeline ends here, but your training doesn't. Come explore or work with us at CruxSci.com.